So good evening, ladies and gentlemen, and welcome to our first ever virtual SETI Talks, which is the monthly lecture series produced by the SETI Institute in Mountain View, California. My name is Bill Diamond. I'm the president and CEO of the SETI Institute. We're a 501c3 nonprofit research and education institution where presently the lights are out and nobody's home, but our science and education and outreach continue. And behind me is our beautiful Allen telescope array up at the Hat Creek Radio Observatory in Northern California. So I'm not actually there, but it's nice to have that backdrop and remind us of a key part of our science. So needless to say, the coronavirus is forcing us all to rethink how we live, work, and function in a highly constrained environment. Tonight's virtual presentation is one example of how organizations can overcome situations like this and unexpected things which interfere with our daily lives and normal ways of doing business, and how we can leverage tools and technologies we're already familiar with to overcome these obstacles and stay mission focused. Our SETI Talk series is usually presented live at SRI International in Menlo Park, California. Each lecture or panel discussion is video recorded and they live on through our SETI Talks YouTube channel. So I hope if you're not familiar with the series, you'll take an opportunity to familiarize yourself with that YouTube channel, which has hundreds of amazing lectures and presentations on our science at the SETI Talks YouTube channel. I often like to ask the audience when we're live and in person, how many are attending a talk for the very first time? So I'd like to continue that tradition, even though this is a virtual environment, and ask you to let us know if this is your first SETI Talks experience, and also where you're joining us from. And you'll see at the bottom of your screen where you're in your Zoom environment that there's a little icon that says Q&A. So you can click on that and you're able to type in comments and questions. Come back to the questions in a moment, but let us know where you're watching from, and also if this is your first time attending one way or the other a SETI Talks. So please take advantage of that on the Q&A platform and let us know where you are. So tonight we're going to explore the world of extremophiles, or life forms on Earth that live in extreme environments inhospitable to human life. What can extremophiles teach us about life on our own planet and the interaction between life and environment? And how does the study of extreme biology inform the search for life beyond Earth? To guide tonight's discussion, I'd like to introduce and turn over the podium to astronomer and senior scientist at the SETI Institute, Dr. Frank Marchese. Frank will introduce our speakers and lead the conversation. And at the end of the formal conversation that Frank oversees, you'll have the opportunity to ask our guests questions and participate in the program. So Frank, over to you, and everybody, thank you for joining us. We're delighted to have you along, and we wish you a wonderful evening and a great session tonight. Frank, over to you.
Thank you, Bill. Thank you very much, everybody, for being here. So if you are here listening to us today, it's because you're probably interested in the search for life in our galaxy and our solar system, of course. So you probably heard about extremophiles, those organisms that can live in extreme environment on Earth, organisms where human and other creatures of our biosphere will not survive more than a few hours. So I am not a biologist, I'm an astronomer. I'm just gonna tell you that if you look around us, just in our solar system, let's restrain the problem to a few planets here, you will see the large diversity of environment in our own solar system. Planets can be very dry, very hot, have a high pressure for the atmosphere, have a high radiation. So it's very likely that if there is planets over there, they will have a different environment as well. So if you want to find life, if you want to know how to look for it and what to look for, you really need to study those extremophiles that can survive in those extreme environments. So to tell us about this beautiful research and stories, I invited those two researchers at NASA Ames. So the first one is going to say, hi, Jon. Hi, Jon, how are you?
Hi, everybody.
So Jon is a scientist at NASA Ames Research Center. His research focuses essentially on enabling human and robotic space exploration and the search for life on other planets and moons. Jon has developed and tested life science hardware and biological experiments on board the Space Shuttle at the International Space Station. Jon has been involved in a large number of expeditions around the world, everywhere. I'm impressed when I look at the list, from the desert of Atacama to Antarctica to Idaho, etc. He's going to tell us about this, of course. Jon is the director of astrobiology operations at the NASA Astrobiology Institute. The second speaker is Jared Broddrick. Hi, Jared.
Hey, everybody. Can you hear me?
Yeah. Jared is a postdoc in the Exobiology Branch at NASA Ames Research Center. His research consists in, I'm quoting him here, applying systems biology techniques to questions of interest to the astrobiology community. In this position, Jared has combined field site exploration, next generation sequencing, and computational biology to characterize the metabolism of microbial communities in the Yellowstone National Park. And you can probably tell us a lot about this. I should mention that Jared is also a lieutenant colonel in the Air Force Reserve, and he provides intelligence support to space operations at Vandenberg Air Force here in California. So let's start with you, Jon. You have a slide, I think, to show us, to tell us a bit about your research and your ideas about extremophile.
Sure.
Can you share this with us?
Let me know if you can see it now. Can you see this? Great. So thanks, everybody. This is a really interesting opportunity. I started my research basically as a farm kid in North Dakota. And I became very interested in how life has the ability to withstand harsh environments, that is the winters in North Dakota. And I became fascinated by things being able to survive in extremely cold environments there and come back year after these terrible winters. And it got me thinking as a young child, is there life outside of Earth, and could it exist, in other planets and moons? And I was a science teacher for a few years, and then I went back to graduate school, and I got engaged in field science in desert and polar environments. And I carried on my experiences here at NASA Ames Research Center, where I've done a combination of both space biology and astrobiology investigations. That is, what happens to life forms when they leave Earth? And more interestingly, in my opinion, is if human explorers can go somewhere like Mars, where should we look and how should we look? And what are we going to find when we explore these very interesting locations? So I've conducted a variety of experiments on the space station, in Antarctica, and also in various desert environments, and including some hydrothermal features here in North America and Idaho. So that's sort of in a nutshell, a combination of both space biology and astrobiology, contributing to both human and robotic exploration.
Thank you. Jared, do you have your slide also can show?
I do. It builds a little bit, so I'm going to maybe mention a couple things first, and then I'll bring the slide up. But I just want to actually start by echoing some of the things that Bill said. About this is definitely an interesting time for everybody, and I really like how the World Health Organization changed it from social distancing to physical distancing, and I think that's apropos when you think about the scientific community and how for centuries has generated social connections and fellowship over long distances. And I'm really thankful for the SETI Institute for inviting me to participate in that long tradition. And so I thank for everybody to participate as well. As Frank mentioned, I'm a self-proclaimed systems biologist, and systems biologists tend to look at life slightly differently than maybe the classical approach to biology, which is maybe to think of an individual component and learn as much as you can about that one thing. Systems biology tends to look at life more as a series of networks. How do those individual components actually interact with one another? And then if you create these different networks, how do different networks interact with one another? And one of the important aspects when looking at networks and their interactions is actually something Bill mentioned, is the concept of constraint. I think we're all experiencing a form of constraints right now with different shelter-in-place requirements or having quarantining ourselves. And so essentially, when it comes to microbes, a lot of times life the constraint actually comes from the environment. And so what I'm going to talk about here is, that kind of leads into the concept of extremophiles. And essentially, so when you're thinking about constraints on life and extremophiles, essentially extreme networks place a constraint on the life that exists there. And extremophiles are just organisms that have found a way to alleviate that constraint. And so, as Frank mentioned, I spent some time in Yellowstone National Park. And here's we zoom in, there's a pretty striking example of temperature as a constraint. And so this white line here kind of elucidates a temperature gradient from the center of this hot spring, Grand Prismatic. It's an iconic location, hydrothermal feature in Yellowstone National Park. And in the center of this, it's pretty warm. It's around 85 centigrade. And there's very little life, if any, that can survive. But as it radiates out and cools down, there's this gradient from this high temperature down to a lower temperature, all the way out to about 30 centigrade on the outside. And these colorful bands that we see are microbial communities that are made up of, the color comes from a dominant microbe, that's a photosynthetic microbe. And these different the color comes from the fact that these microbes use slightly different light-catching pigments in order to perform photosynthesis, and that gives them these different colors. But what's so striking about it is just how perfectly banded these are. And there's a very narrow temperature range where these organisms tend to be happy. And so that's really, it's an interesting, striking example of how temperature can constrain life, and how, this is something John and I were talking about, with really the extremeness of a situation depends on your point of view. For the, you look at the orange guys on the outside, that's a little cyanobacteria called Formidium. And if it gets much above 60 degrees, it's too hot. But if it gets much below 35 degrees, it's too cold. And so really for it, room temperature for us is an extreme environment. And so an important part of when you're looking at extremophiles, is to realize that for the organism that survives in that environment, that's completely normal for it. And so this is really what motivates a lot of my research, is understanding how does the environment apply constraints on life, and then how does life come up with creative ways to alleviate those constraints in order to survive and persist in these environments. And the hope is that through understanding those two facts, we can start to have a better appreciation for how life evolved on Earth, and if what life may look like on other places within the solar system. And an example is home plate on Mars, which the scales of these two pictures are almost identical. And so it's entirely possible that 1 billion or 2 years ago, give or take, on Mars, there was something very similar to Grand Prismatic that hosted some sort of microbial community that was also dealing with the constraints placed on it and trying to find a way to thrive. And so that's kind of how, that what motivates me and what I'm kind of interested in researching.
Thank you.
If I can give this back to you somehow.
It's okay, we can keep this pretty picture of the Yellowstone here. All
right.
You didn't mention, let's just start with you, Jared, because you didn't mention what basically pushed you to this research. What is the, what was the motivation for this kind of research? Where did you get this idea that extremophiles are interesting and you wanted to basically dedicate your career to study those?
Well, I think that.
So, oh, no, John.
Sorry, go ahead.
No, please, John. I'm going to drink my beer.
I just have been interested in understanding if life can exist elsewhere, and wouldn't it be fascinating if we can discover life existent, or the evidence of past or present life on another planet or moon. And I became most interested in Mars because it's probably the closest terrestrial planet that has an atmosphere that humans might be able to use or be able to explore themselves. And so I became interested in exploring places that are probably similar to where, if life existed on Mars, where it might have last existed or still exists today, if it in fact does exist. And those two places primarily are either ice or salt-rich environments. And ice or salt-rich environments are thought often by astrobiologists as analog environments for ancient environments on Mars. And so if I want to explore places on Earth that are sort of simulate some aspects of what ancient Mars might have been like, I need to go to the coldest places on Earth, or some of the driest places, or some of the saltiest environments on planet Earth. And so my explorations have taken me to a variety of these locations. And I, much of the time in the early expeditions that I was carrying out, I was imagining myself as an astronaut exploring these landforms and these features, and imagining myself carrying tools that an astronaut might be carrying. And one of those tools that I was carrying was a micro R meter. And I was interested in using a micro R meter because I could get a radioactive signal from an energy-rich rock, and perhaps that energy-rich rock might be useful for people in terms of energy production. Well, on about 5 or 6 different expeditions, including Antarctic and the Middle East, I carried micro R meter on all these expeditions, actually trying to take measurements and looking for a radioactive signal, and I didn't see anything, except one particular expedition where I was at Warswick Hot Springs in Idaho. And what I'm going to do, I'm going to share my slide back again. Are you able to see the slide?
Yeah.
Great. So in the upper right, you can see 2 individuals there taking some measurements and data from a small hot spring. And this is sort of a very small version of what Jared was talking about with the giant hot spring in Yellowstone. And we see similar features here where there's a lot of silica-rich deposits that are also seen at home plate. And at this location, the micrometer was going off, and I discovered that these hot springs were radioactive. It was completely by accident, where I had discovered that this particular site in Idaho had a slightly enriched radioactive signal. And we're talking like 4 to 5 times background. So we put together a proposal, and we studied this environment for about 4 years, and we learned about the microbes that lived there. And I was primarily tasked with studying the environmental conditions, including the temperature of the waters and the nature of the radioactivity. And what I discovered was that as the temperature was increasing, we saw an increase in the radioactivity signal as well. We didn't expect that. So there seemed to be a correlation between radioactivity and temperature. And we did also identify and see that there was some small amount of hydrogen being produced there, whether that was from somewhere in the Earth or from the photolysis of water from gamma rays and X-rays, splitting water to produce hydrogen. Fact is that it was there. It got me thinking, wouldn't it be interesting if there was energy-rich rocks that contained thorium on Mars? And I went back to some old maps from the Odyssey mission that mapped the thorium in parts per million in the near surface crust of Mars. And lo and behold, there are areas on Mars that are thorium-rich. And so I became very interested in whether or not these thorium enrichments, and it's only about 2 to 5 times background in some of the lowest signal areas. The fact is that there's an increased energy abundance in these areas. And I'm still to this day looking to explore whether or not enrichments in thorium in rocks either contribute to habitability or somehow make it less habitable, or do damage to molecules that might be in those locations as well. So as much as a component for habitability, I'm also interested in the thorium signal in this area of Mars as a exclusion criteria for a particular landing ellipse if we're looking for signals of evidence of past life. And so it's interesting, a side note, some of these thorium-rich regions on Mars actually are also enriched in ground ice, which is an interesting topic in terms of potential habitability. And so the connection to the thorium and the hot spring in Warswick, I continued my explorations in the Atacama a couple of years ago, where I was working, and you can see in the bottom picture there, with the ARADS exploration team, the Atacama Rover Astrobiology Drilling Studies team. And there are several pits that have been dug where the rover that's designed to detect evidence of present life forms, either forms of antigens or amino acids. And I also made some measurements there in the Atacama that suggest that there's elevated radioactivity in those soils as well. So I'm trying to make a connection between the thorium that we see, thorium and uranium that we see on Earth, and the thorium or uranium that we might see on Mars as a potential beacon, so to speak, of where to dig or where not to dig for evidence of past or present life. And I've carried out a few other exploration and expedition-like activities in Antarctica, where we've tested some simulated, some prototype spacesuits technologies, where we simulate operations that mock up data sample collection and data collection, so that we can practice and learn what are the things we need to do once we get to these very hostile environments. And so in a handful of what's considered extreme environments here on Earth, I'm sort of practicing both with robots and humans what it takes to explore these locations.
Thank you. What about you, Jared? What's the motivation behind your research and your personal motivation as well? I'm not only the scientific one.
Yeah, so I mean, my motivation is, I've always been very, very much inspired by it and motivated to participate in the NASA mission in general, and human exploration of the solar system in particular. I took a pretty roundabout way to get to NASA. I did my undergrad at the Air Force Academy, and I was commissioned in the Air Force for a couple of years full time, before I realized that I mean, science was where my heart was, and so I left and pursued that for a bit. I came back to get a PhD rather late in my life. I was about 33 when I started up again, and I had a 11-month-old son at the time, which was kind of poor decision making, to be perfectly honest. And so I was forced into, I started out trying to do classical wet lab biology and realized that that wasn't going to work with a 1-year-old at home. And so I dabbled in computational biology, and I was fascinated by it because it essentially allowed me to ask some questions that I'd always was interested in. I was always trying to come up with causation. Causation in biology is a really, really dangerous game to play, because it's the problems are multivariate. There are a lot of interacting parts, and so it can be dangerous to try to really draw a straight line between what you observe, the phenotype of an organism, and some kind of causal stressor or event that drove that phenotype. But with computational biology, in particular, the type of biology I studied at the University of California, San Diego, in the Systems Biology Research Group, was this concept of constraint-based metabolic modeling. So how does external constraints on a living organism's metabolism change its phenotype? And it was eye-opening, because it was, look at some of the research done out of that lab and others, and you have these experimental evolution series where you'll put E. coli or some other bacteria under a constant stress, and you'll watch it over time. Its growth rate increase, its growth rate increase, its growth rate increase. It is solving the problem of that stress. And then you can go back in and you can sequence the genome, you can collect data, and using these techniques, you can actually draw a straight line between a single mutation in that organism and why it was able to all of a sudden achieve a higher growth rate, why it was able to all of a sudden become more fit under that stressor. And it was just, it was such an elegant way to look at biological problems. And it really, and so when I came back to NASA towards the end of became interested in coming back to NASA, and I was starting to look at where in the NASA portfolio with this line of thinking and these methods most applicable. Astrobiology and space biology were really the places where it was at. And I've always had I studied photosynthetic microorganisms in my PhD. And so looking at things like photosynthetic microorganisms and hydrothermal systems is a pretty it's not too far of a stretch to extend that initial understanding and effort into something that was much more applicable to NASA's goals and interests. And also, I mean, how many people get to say they're paid to think about the origins, evolutions of life on Earth and possibility of life elsewhere in the solar system? It's just a cool way to spend your day. And so that's exceedingly motivating for sure.
Thank you. So before we continue this conversation, I would like to mention we have almost 200 people watching us at the moment, and they send us their location. So we have people from St. Paul, Minnesota, Mapleton, Philadelphia, Toronto, Sydney, Australia, Robbinsville, El Cerrito, California, Madrid, Spain, Michigan, in Cleveland, in Pakistan, New York City, and Victoria in Canada, and Copenhagen, and also Mauritius, an island close to Réunion Island in the Indian Ocean. Hello, everybody. Thank you for following us. So don't forget that you can ask questions as well by clicking on the Q&A here. We have 2 moderators that will take over your questions and send them to us in a few minutes. So if you have questions during the conversation, please write them down. We are paying attention. Thank you. So I have a question also for you, Jon. You mentioned experiment, and you show one, for instance, that you have done in Chile. People are always curious to know how biologists truly work, what instrument they use. You describe one of them, you describe the measurement of radioactivity as well. But can you give us kind of an overview of the type of measurements you do when you're on the site, and the type of what you're exactly searching when you analyze those rocks and when you look at those environments?
Sure. It kind of depends on the nature of the investigation of the experiment. For example, you might, in culture studies, you might do a variety of cell counts where you're counting cells that are growing in culture or how rapidly they're growing. In mineralogical studies, you might use X-ray diffraction to identify particular types of minerals. And in the case of other types of biological studies, such as the solid experiment that was on the rover in the Atacama, that uses a antibody-antigen complex reaction to identify the presence of antigens in the environment. So we're actually combining different types of biomolecules that are found in the environment with antibodies that are consistent and designed to interact with them. And the combination of those 2 molecules is what will produce a signal that will indicate the presence of a biological molecule. And in the cases of trying to identify different kinds of microbes, we'll use genetic sequencing to be able to determine the nature of the organisms, specifically what kind of species there are. And you might even use other technologies such as the MinION, which is a nanopore DNA sequencing tool that can be used in the field to actually sequence DNA or RNA right there on the spot. So these kinds of technologies are what we're working with in terms of development for flight instruments to be able to actually take those measurements on other planetary surfaces. So imagine taking a MinION that's developed on Earth, repackaging it for flight, and then sending it on a rover to another world to look for biopolymers in the dirt on other planetary surfaces. So that's an example of some of the technologies that we're working with.
Thank you. So Jared, I'm noticing you're very interested in studying those as a system, basically, and studying the interaction between them, right? You show on this environment and this picture, these different colors coming from different type of extremophiles. I always wonder, and I'm not a biologist, so I really ask you this question, and I'm hoping that you are not making you are not going to consider that I'm completely ignorant, but I'm always wonder, is there any common, when you study an environment like that, with a species which can survive at different temperature, for instance, is there a common genetic pattern, is there any connection between them that will explain how they survive in this environment and how basically they thrive there and they're all there together? Is there any way they communicate, not communicate, but they basically kind of are cousins or into an evolution, or they are very different and it's a coincidence they're here? How does that work?
Yeah, right. So it's there's two different orthogonal directions to that, they're kind of right angles to each other. So one is, in general, those are all different species, and so their genetic background, their history, their evolutionary history is very different from one another. But the other argument there is that in general, for a given stressor, there tends to be a lot of similarities in how the organisms overcome that stress. And so temperature stress is fairly is well studied, and it typically, you have to come up with ways to change the proteins that are in the cell that kind of perform the essential functions have to be adapted to function at a different temperature. And that usually that adaptation process is you change some of the amino acids in the protein that makes it more stable at higher temperatures. You think about boiling an egg. I mean, that's the albumin of an egg. Once it gets to a certain temperature, it completely turns into a solid, and therefore it's completely changed at that point. And you don't want, if you're a cell and you have proteins that perform functions, the last thing you want to do is overheat yourself and then all of a sudden turn into a boiled egg. And so the strategies that are usually are somewhat conserved. There are, of course, that's an existing line of inquiry in a lot of the different types of stressors between salt tolerance and temperature tolerance. But in general, when you look at thermal tolerance, it's really, it has to do with changing the amino acid structure of the protein so that they operate at higher temperatures. And basically what happens is, as the organisms are getting, encroaching into a higher temperature, the first organism that, based on luck of the evolutionary draw, has that mutation that allows it to go into a higher temperature, it kind of claims, it plants a flag in the ground at that higher temperature and thrives. And that tends to exclude anybody else that's trying to get in there, because at that point, it's already consuming all the resources available for any newbies to come in. So which is kind of maybe part of the reason why you see such a dramatic separation, is that once somebody gets that tool set to go into that higher temperature, it has that temporal advantage. It has time and space to occupy that area, which makes it more difficult for the next guy coming in to try and occupy that space. So
just to continue in this topic, if I take a hot spring in Chile and a hot spring in North America, the extremophile living on those will be identical, or they will be completely different, and that's just an evolution that?
Very different. It'll be very different depending upon the nature of the water that's coming out of the hot springs, or the nature of the rocks that are present near the hot spring, or what the source of the heat and how that is influencing the environment. So there might be similarities in some cases, but in others it might be completely different.
So the species are different, but the way they manage to survive in the heat, it's the same. Or even this could be different. The survival strategy is different.
So I think it depends on the stress. But no, in general, it's actually quite fascinating. I've been doing a lot of sequencing in Yellowstone, and these are not well-characterized organisms, but you take the DNA sequence and you toss it against the database, and the hits come back, and there's very similar, to the species level in places like, I'm in Yellowstone, in India, in Western Europe. And so even at the species level, it does seem like the tool sets that the organism brings into that environment can give it kind of an advantage, and that seems to be conserved. And so to Jon's point, really the hot spring itself, you got to think about the hot spring is not a hot spring, not a hot spring. And so the pH is different, the temperatures are different. Depending on the rock that the water is interacting with, it pulls out different minerals, and that completely changes the chemical composition of the fluid that's coming out of that vent, out of that into that hot spring. And that different chemical composition is a different constraint. And the organisms that, when the organism encounters that constraint, whoever figures out how to overcome it first wins. Sometimes the strategies are the same between different sites, or sometimes it's unique. And so it really depends. That's why Jon can traverse halfway around the world in different places and find so many different unique strategies for things to survive, even though that there's the constant features that it's there are some constant features, but there's certainly a lot of interesting differences to explore.
And each of those differences will alter the biology and its ability to be preserved or be destroyed in the rock record, whether it's sinters that are formed as evaporites around hydrothermal environments, or the different types of biomolecules that might be preserved. For example, one of the, we've talked about proteins and genetic material. Another biomarker that we're interested in are lipids. And lipids are one of these molecules that make up cellular components, and they seem to be something that can be preserved for very long periods of time in the geologic record here on Earth. And so we would expect that to probably be true elsewhere if life could exist elsewhere. So if we're looking in really old ancient rocks, the biomolecule that we might be best suited to look for are those molecules that can survive the longest in rocks, and that might be lipids. So some of the technology development that we're working on to look for evidence of ancient past life are specifically designed to extract lipids from rocks.
So that's an interesting point. It means that it implies that life can, there is different way for life to thrive in extreme environment. There is different species, but your job is very complicated then, because if you want to find biomarkers, you really need to understand the details of how this life developed in the extreme environment. And from the conversation so far, I have a feeling we don't really know yet a common biomarker for all extremophiles. It's not that simple, right? You need to basically look for different type of extremophiles. You basically try to find a biomarker, but they may be completely different. So how can we apply this research for the search for life on other planets then? Do we always have to go back to the extremophile on Earth and make some kind of comparison? How do you envision this?
Yeah, so that's exactly the type of research that's going on right now. And the main challenge we have is that a lot of, especially before the sequencing era, a lot of biological research was focused on a small subset of all the organisms on the planet. And you get into these environments now, and you're seeing just some amazing, amazing corners of the phylogenetic tree, where these organisms are doing some really interesting biology that we just don't know anything about. But to Jon's point, it's really a relationship between the type of biological material and then it's also its persistence in the rock record. And biology takes things and puts them together in some pretty interesting ways that has a pretty strong signature. And what's really cool about hydrothermal systems is that not only do they give you the ingredients for life, which is all of those minerals and metabolites that get leached out of the rocks as the water comes to the surface, but it also, the mineralization preserves the biomarkers quite well. It kind of encrusts them in these mineral protective mineral casings that is just sitting there waiting for us to discover. Going back to the image I had of Grand Prismatic, there was an astrobiology research out there about 10 years ago now, where they cut vertically into the rock, and in the top you would have the more classic biological signatures like nucleic acids and maybe some proteins, and as you move down you may get some of the photosynthetic pigments, and right down at the bottom, several centimeters down, the only thing you could really see were the lipid biomarkers, and those are pretty. Those are like compounds with 30 carbons in them, all connected in very interesting fashions. It's very. I do not personally. I would stake my biology claim in that nature wouldn't be able to produce a 30 carbon ring structure that you would see in these lipid biomarkers. And so they're pretty attractive chemical to look at when you're looking at for life.
All right. So I read somewhere that people, it's kind of another topic for the conversation, but some people have been taking extremophiles on Earth that can have the potential of living under extreme temperature, for instance, and they've been trying to see the limit of in temperature of this extremophile to kind of get on a range of possibility for exoplanets habitability, for instance. Have you done this kind of study, and what can you give us some examples of study similar to this, for instance?
So I could jump in there, but this would be looking at it from a space biology perspective. One of the things that space biologists are really interested in is the influence of gravity on living systems. So one of the things that's pretty cool about the astrobiology of Earth is that gravity has been more or less constant for the entire existence of life on this planet. And understanding the relationship of how gravity influences living organisms and if or how it might influence the origins and evolution of life, space biologists take their experiments from Earth to the space station, for example, where they are in free fall, and they're able to study biological functions in a zero gravity environment. So we consider that to be the spectrum of zero gravity, whereas on Earth, we can study life forms in 1G. And then we can also study life forms in hypergravity, which is greater than 1G in centrifuges. We can also study life forms in centrifuges on the space station, where we can get an acceleration that's slightly more than zero, but less than Earth. And so seeing biological responses across that spectrum helps us understand the biological influence of gravity, or a spectrum of gravity, on living things. That's one example.
Thank you. Okay, I think we are ready to take some questions. And if you're ready, Rebecca, you just pop up, so I think you're ready.
I am ready. Hi.
Yeah,
there are a lot of questions, and I don't know that we're going to be able to get to all of them, but the first question comes from Nicole, who asks, can you explain the best evidence for extant life on Mars?
I'll take a shot at that. There's no evidence of life at this time anywhere else in the solar system, or anywhere else in the universe for that matter. So we are able to say that we do not know or see that it is present. Jared, do you have any thoughts on that?
No, and it's of course the most enticing question, but if you want to say evidence, I mean, no joke, evidence. I think we're actually trying really hard to just establish what are the tests that you could actually be definitive that yes or no. I don't even think we currently have a really good protocol on even answering if we see something that we could definitively say yes or no. And so that's part of our research as well.
Yep.
Okay, I'm going to take another question.
Okay, let's have the list next down the list. This is a question for Jared. Could the organic bands in the prismatic pool be due to a lack of competition? Effectively, the predators cannot enter the hotter region. So this is a predator-prey question.
Yeah, it's the concept of predator-prey, I think, is very much relevant to that environment, and part of why I think these organisms migrate into extreme environments is because of a lack of competition. Essentially, especially in these situations where you might be resource limited, if you can stake a new claim in a slightly extreme environment, you're the only one there, then you don't have much competition. And so that's definitely a portion of the dynamic in that is, now it's another aspect. Well, that's probably good enough. I'll leave it there.
Thank you.
Okay, the next question comes from Andrew. Using the example of the extremophile organisms at Gram Prismatic Spring, is it the trend for all extremophiles to thrive within a certain specific environmental range, or are other extremophiles not so choosy?
So for temperature dependence, it is very strict. Essentially, by migrating to a higher temperature, you've now relinquished your fitness at a lower temperature, and that is a truth. You get into things like halophiles, things that like salt. I don't know if that holds quite as true, because it depends on the strategy that the organism has had to acquire in order to survive in that environment. Going to temperature, you're fundamentally changing the structures of your proteins, and they're only going to function in a very narrow temperature bandwidth. And so if you move out of that bandwidth, the organism's not going to be too happy. Now, if it's something where it's detoxifying salts by using energy, then maybe by going to a lower amount of salt, you may actually free up energy that you don't have to spend to try to survive in that environment. So in that situation, it may actually be, it may be a fitness advantage to move back into a lower salt condition. But then again, relevant to the previous question, the competition is probably higher in that lower salt condition as well.
So what about, John, what about radio-resistant?
Yeah, that's a good question. I was just about to jump in on that. If I'm not mistaken, we see that desiccation resistance and radiation resistance are somehow linked, and that if you have one, you have the other. So I haven't done a lot of research personally in that area, but I've read a fair number of papers about that. And it's very interesting that microbes will develop this radiation tolerance similar to saline tolerance, if I'm not mistaken. I don't know if it's vice versa, but I know that if you get one, you get the other. Jared, do you recall any of that in any of the work you've done?
I haven't seen anything on that.
Thank you. Simon,
okay, it's a question about life on Enceladus, which is a moon of Saturn. If Enceladus has a liquid ocean under the surface, are there any extremophiles that could survive in that environment? And that's from Dan.
So I love this question. Enceladus is definitely a hot topic right now, and the liquid ocean definitely is enticing for the possibility of extant life in our solar system. It's important to kind of caveat that with the fact that if the only kind of energy available there is geochemical energy, which is pretty low yield, and so you wouldn't have a really, I mean, some of the calculations I've seen, you're not going to have a super vibrant biosphere. But it's also, this is something, I don't think we really have time to talk about this too much, but there's a big difference between the types of environments which life can emerge, like it actually have a genesis of life, and the types of environments which life can evolve into. And just because we have life in cold, saline ocean waters on Earth doesn't necessarily mean that life can emerge, or actually the origins of life can be in the same environment. And that's a completely different topic, but it's an important distinction that just because we see life in an environment doesn't mean life can actually emerge in that environment.
So this is an interesting point. That means that because we have life on Earth, life diversify. So there is a way for this life to adapt to an extreme environment. But on Enceladus, if we assume that Enceladus never had any connection with Earth, life will have a hard time to diversify on this small body because there is no change of environment. That's basically what you mean, or why the life on an Enceladus will not diversify to the point that it will survive the environment on Enceladus.
So we do not fundamentally know the conditions under which life can emerge. But by conjecture, just looking at basic chemistry and thermodynamics, that the more extreme the environment, the more limited the chemistry that can occur. And so the emergence and the genesis of life, a lot of the theories are it's going to be somewhere between 20 and 50 degrees centigrade, just because a lot of the biochemistry that we know of on Earth is kind of relevant in that range. Now, if there was a genesis and there was something similar on Enceladus, and there was something similar to Darwinian evolution, there's nothing that would necessarily stop a more diversification. But there does seem to be a connection between the diversity of life and the amount of energy available to life. And really, you saw in the history of the Earth, we saw the phylogenetic tree really explode after the oxygenation event. Once oxygenic photosynthesis occurred, the diversity of life just exploded, because the energy available to the planet just as a whole was completely different. And so if you're thinking of a small icy moon that's only getting its energy from a little bit of geochemical activity, there's just not going to be a whole lot of excess energy in the system to maybe provide that kind of diversity that we might see on Earth. Now, that I could be completely wrong. I'm basing all of these assumptions on life of the N of 1, which is what we see here. But I know Jon has looked a bit on, especially looking at kind of gradients of stressors, and there might be some interesting anecdotes he has from that research. So I yield to him.
I would echo everything that you've said, Jared. What I was going to say is echo the other point that you made earlier in the evening. Was it sort of depends on the point of view of is it the extremophile or the organism in the environment. And I would argue that if we took a human and placed the human on Mars, we would be an extremophile on Mars. But we would have been required to develop a technology to keep us alive, to put us in that environment. But any Mars organism that would be looking at us from their point of view would sure think of us as an extreme environment dwelling type organism, because we require this oxygen rich atmosphere that is completely foreign to a Mars environment. So, and even if there was no life on Mars, if we were able to transport life to Mars, here again, it didn't originate there, but we would be able to take it there.
Thank you.
Sorry, okay, the next question comes from Frederick, and it could be for either of our panelists or both of them. What research, discovery, or insights have very much surprised you in the past 6 to 12 months?
Jon, you want to go first?
Well, I guess the research that I've been looking at most closely is sort of the virology of what seems to be captivating the Earth, and that's while that may not seem like an astrobiology topic, I would argue in some ways that it is, because astrovirology is actually a very, very important topic as it relates to the origins of life or the evolution of life on planets. And we have an example of how a virus has moved throughout the entire biosphere very, very rapidly, and how it appears to be changing, and the nature of how it is influencing the life forms that it's infecting. We're learning a lot about what it does, what it doesn't do. We're also learning a lot about how to respond to this kind of phenomenon. So that's the kind of research that I've been looking at most deeply in the last 2 weeks, and I really haven't dug too much deeply in other areas since that.
What about you, Jared?
I was able to sequence a bug in Yellowstone that is completely different than, it was basically everything about it is different than what we expected from this particular species. And so it's pretty cool to be able to find something new and explore it. So that's been pretty exciting.
Did you publish this already?
I'm working on it.
Did you name it?
I'm going to name it after my dog. So, but I'm not going to tell you the name because that's going to give it away, and I don't want to steal it from me.
Simon, do you have a question?
Yeah, actually, it goes back to the concept of maybe the virology. This is from a medical student. What are some of the applications of astrobiological research to help advance human health here on Earth? And that's sort of being touched upon just now.
Astrobiology for human health. Well, Uh, I guess it depends on if it's a pure science topic or an exploration topic. I think astrobiology and space biology and human research really go hand in hand, especially as it relates to human exploration of astrobiologically interesting locations. From an astrovirology perspective, there's probably overlap here that we might be able to explore in a synergy of astrobiology and space biology, and that is when humans go into space and experience the conditions of spaceflight, our immune systems are altered and our body starts to respond similarly, very much like what Jared was saying, how microbes will respond to the stressors that they experience in hydrothermal environments. Humans are doing the same thing in spaceflight. And the viruses that are in our bodies that we take with us during spaceflight are changing as well in their ability to cause a biological response. And we see reactivation of some viruses in astronauts. While we haven't had any major issues in terms of human health, being able to respond to a more virulent condition, whether it be through drug therapies or through different types of dietary measures, are the things that the human research program is very interested in as it relates to viruses and short and long-term human health and safety of missions. So that's one example.
Thank you.
The quick 30-second anecdote. As a medical student, they're probably aware of the concept of polymerase chain reaction, PCR. It's a technique used in molecular biology and used even to test for the coronavirus. It requires an enzyme in that kit, and that enzyme was actually taken from a thermophile in Yellowstone National Park. That's where it was, because it can tolerate high temperatures, it can be used in that polymerase chain reaction. And so there are a lot of biotechnology applications that can be taken from these thermophiles whenever they try to find a way to alleviate a constraint.
Thanks.
Next question comes from Scott. To what extent do you explicitly try to match up extreme environments on Earth where you do research and environments on planets we know about? I heard some of that regarding Mars, but what about Venus, Europa, other moons? Does this way of thinking even make sense for the gas giants?
So John and I talked about this a little bit, the concept of analogs. This is when we're talking about there's almost an analog trap where you think of something similar and therefore it's completely applicable or directly applicable. You have to be very judicious in the application of these understandings and really ask the question, which variables are going to be consistent between the two, and really only focus on those variables. Otherwise, you're going to be overstating your case quite a bit.
Yep, I would definitely echo that as well. And perhaps another way, rather than traveling to an analogous environment that you think might be similar to an ancient or present day location on some particular planet or moon, it might be more accurate to physically simulate the exact conditions in some type of simulator that might simulate atmospheric pressure and temperature and atmospheric composition, such as in the higher altitude ranges or mid ranges of Venus's atmosphere. And as just a side note, I'm very interested in aerobiology here on Earth, and also the exploration of Venus's atmosphere to look for evidence of some type of biology, if it could ever exist there as well. Chances are it's too hot on the surface, but we should look and figure it out before we say definitively yes or no.
Okay, thank you. Simon, do you have a question, I'm assuming, right?
Yeah, this is perhaps a planetary protection question. This is from Surav in Malaysia. Is there any possibility or proof that virus or bacteria can survive in space from other solar system planets or even exoplanets? So I suppose in terms of getting them through space and maybe returning to Earth. I'm extrapolating the question there.
Sure. Yeah, this is a very interesting topic. What's fascinating, at least from my perspective, in the last year or so, we've seen two interstellar objects pass through our solar system. And because we know that rocks can travel from planets to planets here in our solar system, you can imagine that that rock might have been part of another planet somewhere else. And so then you begin to imagine, well, could those rocks carry life forms, or could they carry fossils of life forms? And the answer is possibly, but of course we don't know. But Earth, like all the other planets, is constantly experiencing sort of a constant bombardment of micrometeorites and larger objects falling through the atmosphere, and some of which burn up, but others hit the oceans or hits the Earth. And so we're constantly gaining weight here on Earth from rocks outside of planet Earth.
Okay, I have a final question for both of you. So you describe your current research. I'm kind of interested to know what's next. What's next for each of you? What's next in terms of what's your next big project, your big question you want to answer to? Or what is the next place you want to explore on this planet or elsewhere?
Well, I need to do some writing and get some manuscripts published. That's job number one. I was brought in on the NASA Astrobiology Institute to merge it with the astrobiology program, and I would like to work on developing synergies now with the astrobiology and space biology worlds across NASA, and so I'm going to be focusing a lot of my efforts on that. But I'd also like to continue to do some exploration of desert and polar environments, and I've got some sensors in the ground that I put into the dirt in the permafrost in Marambio, Antarctica. It's an Argentine military base. That was a few years ago. I'm sure the batteries are dead, but the data's there. And I'd like to go back and collect those sensors and see how that permafrost has changed in terms of its temperature. Is it the same or has it warmed?
What about you, Jared?
So, I mean, similar to some of the Enceladus questions, I'm interested in taking some systems biology techniques to undersea deep water vents and looking at some of the more interesting seafloor hydrothermal systems that we've seen. And there's some evidence of some very simple primordial metabolisms down there. And I think characterizing those things from a systems biology perspective might provide some hints as to how simple organisms can live on very low energy, like we might see on Enceladus.
Thank you. So, Bill, you want to say a final word? You're mute.
Here we go. We have the technology. Okay. Well, listen, first of all, I want to thank Jon and Jared so much for joining us this evening and sharing their work and their perspectives. This is an amazing topic. It's actually a timely topic. Some of the questions delved into this issue of the relationship between the current pandemic we're experiencing with the coronavirus and the nature of viruses in general and how they relate to extreme biology, which they do. So it's been an interesting topic to explore, and we thank you both very much. We thank Frank for moderating the discussion and leading us through this conversation. Speaking of extreme environments, I couldn't help but notice that Frank is in an environment which, as the evening has progressed, has gotten darker and darker and darker. So when we come back next month, we're going to get a light for Frank and his extreme environment and see if we can light him up so that he's not struggling in the dark. But again, this was a bit of an experiment for us at the SETI Institute to do our SETI talks virtually through Zoom. We hope you enjoyed it. We'd love to get your comments and feedback if you have suggestions. We will be back next month. And I think the date, Rebecca, you're going to have to correct me on this. I think the date is April 22nd. Is that correct?
That is correct. The date is April 22nd, and we will also be sending a survey to everyone tomorrow morning. And if you could provide any feedback on how things went well, maybe how things could be better, it will really help us improve and optimize this experience.
That's great. So yes, please do that. So yes, we'll be back on the 22nd of April, and at that time, we'll be talking with Alex Polak and Andrew Simeon and the team we have up at the Hat Creek Radio Observatory and some of our colleagues over at UC Berkeley and the radio astronomy group there, who will talk about SETI, the search for extraterrestrial intelligence using radio telescopes, the radio telescope array, and talk about the science of SETI endeavors. So that should be really interesting, certainly right up our alley, right in the strike zone of the work we do at the SETI Institute, as is astrobiology and the study of life in extreme environments. So please join us for that in a month. And in the meantime, I can only urge all of you to stay healthy, to follow whatever guidelines your local or state authorities are passing on. We are definitely in the midst of a very serious situation that should be taken seriously. And most importantly, we want everybody to be safe and to be back again with us next month for the next episode of SETI Talks. So from the SETI Institute, not in Mountain View, California, where the lights are out, but from virtually everywhere where the lights are on and we're still operating, we bid you all good night. We thank you very much for joining. We thank John and Jared again, and Frank, Simon and Rebecca, and Lee for putting all this together in terms of the technology. So we're very grateful to all of you. Have a safe and healthy evening, weekend, and until we see you again. Thanks everybody, and good night from the SETI Institute.